surprising to see that even after completing masters and PhD from IITs, many scholars are willing to accept low-paying job in private industries.
நீ தரமாட்டுற மைனாண்டியா
reality.
ஆடியோ இன்புட் கேப்சர்
இது என்னதுயா? இதான் வந்து சால்வ்
ஆமா
இது என்னது? இதெல்லாம் வாங்கி வச்சுக்கிறியா தனியா?
ஆமா பஸ்
சரி எனக்கு 10 நிமிஷம் இருக்கா?
Good morning, everyone. It's good to see such a huge number of audience today with us. So right now I can see the number of participants has gone up to 103. That's amazing. Good morning, everybody. Am I audible, everyone?
Oh, yes, sir.
Thank you. Good morning, and welcome everybody to this weekend morning. And here we are going to have a full day packed with knowledge, information, and some fun as well. So I hope all of you have planned your day accordingly, that you are going to be with us till 3:30, 4 in the evening. Okay, if my volume is coming low, then you might have to adjust your system volume. Please check that. Everything is normal. Okay, so everybody else is saying that it's all good. All right. Okay, so good morning everyone once again, and on behalf of the organizing team and the entire team of Biotechnica, myself, Tithi Saha, I would like to welcome you all warmly to today's workshop on genomics, clinical research, and artificial intelligence, pathways to oncology. So cancer, as you all know, is not just a single disease, it's a collection of disorders which are driven by genetic alterations, environmental factors, and complex biological processes. So over the last 2 decades, genomics has transformed our understanding of how cancer develops, progress, and responds. And now, with the rapid integration of artificial intelligence and multi-omics approaches, we are entering a new era where precision and personalization are no longer futuristic goals, but emerging realities. Today's session are carefully designed to take you on a journey from molecular foundation of cancer to the clinical applications of genomics, and finally towards AI, that is artificial intelligence-driven innovations and future career paths. So right after my introduction, we will start our sessions approximately around 10:10. We will be starting. So our first session will last until 11:15, and that first session will be led by Mrs. Divya, and she will set the stage with the genomics blueprint of cancer. So you will explore how mutations in oncogenes and tumor suppressor genes, they actually drive the tumorigenesis. Discover cutting-edge tools like NGS, CRISPR, and single-cell sequencing, and see how precision genomics has actually revolutionized breast and lung cancer treatment. This session will also give you a glimpse into exciting frontiers like liquid biopsies and real-time tumor monitoring. After that, our next session will start from approximately 11:15 and last till 12:30. And that second session will be taken by Ms. Kavita, and she will bridge the science with practice by exploring the clinical impact of cancer genomics. So here you will learn how genetic testing aids in risk prediction and personalized medicine, how genomics-driven trials are reshaping drug development, and the ethical considerations that ensure responsible use of this technology. You will also get insights on how pharma industry and hospitals apply genomics in real-world decision-making. After that, we will take a short break of 30 minutes for lunch. So 12:30 to 1 will be your lunchtime. And again, we will come back and meet here at 1. So 1 to 2 will move with session 3, where Dr. Nilofer will guide us into the data-driven revolution with artificial intelligence. So you will see how machine learning predicts cancer progression and drug response, how it accelerates drug discovery, and how AI is integrated with multi-omics to provide holistic insight into cancer. Importantly, this session also highlights the challenges like bias, interpretability, and validation that must be addressed to make AI critically reliable. And after that, we will move on to our last session. So the teaching session from 2 to 3, which will be taken by Mrs. Somrita, and she will focus on the future careers in cancer genomics and AI. So you will gain clarity on the diverse job roles across academia, biotech, pharma, clinical research, and AI-driven startups. The session will also highlight essential skills ranging from wet lab techniques to computational analysis, and also guide you towards certifications, internships, global opportunities to strengthen your career prospects in this particular field of study. And as I told you that after that, we have kept some fun activities and interactive session for you. So finally, we will conclude with session 5, which will have an interactive quiz and QA session where all our faculty members, all the participating faculty members will be joining, and you will get a chance to ask your direct queries and questions to them. Okay. So everybody, as we progress through the day, please keep writing notes. Okay. So all of our facilities will be available for the QA session. So you write your notes, you write your doubts and questions, and please come up with your question at the question answer session. Okay, so actually what happens is, because we have a very very packed day, so we do not extend any session, because otherwise everything will get rescheduled. So that's why questions, it will be more preferable and more better, okay, if you all can ask your question at the end, okay, in the QA session to all our faculty members. So all our faculty members will be available at that time. Okay, so write your questions, be ready with them, and ask them in the QA session. And before that, before the QA session, before the interactive session starts, we will be having a short quiz as well. And I will tell you all that the winner of that quiz will get some amazing prize from Biotechnica. Okay, so keep listening, everyone. Keep attentive, stay attentive during the sessions, and so that you can get a chance to win that prize. Okay, so in short, today's workshop is designed to give you a 360-degree view of cancer genomics and AI, from basic science to clinical translation, from research innovations to career opportunities. So we encourage you to be interactive, ask questions, and think critically about how this field will continue to evolve. So without further delay, let's begin our journey into the world of cancer genomics and AI, where biology meets technology and research meets real-world impact. So for our first session, I would like to welcome Mrs. Divya. Divya ma'am, can you hear me?
Yeah, hello ma'am. Good morning everyone.
Good morning, Divya ma'am, and we are waiting eagerly for you to start your session and to give us the idea of what is cancer, what all recent things are happening in cancer genomics field and all. So before we start, let me just give a brief introduction of Divya ma'am to all of you. Many of you might be already aware with her. So Divya ma'am is an academic support specialist at Biotechnica from last 7 to 8 years, and she has a strong academic foundation in plant biotechnology and molecular biology. She holds over 2 years of research and mentoring experience from University of Kerala, where she guided both postgraduate and undergraduate students through their dissertation work. She secured an All India rank of 20 in CSIRNet Life Science examination in 2018, And as I told you, past 6-7 years, she has been contributing as an academic support specialist at Biotechnica, helping students and researchers excel in competitive exams and research domains critical. So over to you, ma'am. Please get started with the session.
Thank you so much, Tithi ma'am, for that wonderful introduction. So I'll just share the screen, everyone. Just wait for a minute. I hope so my screen is visible to all of you. Please confirm.
Yes, ma'am.
Okay, thank you for confirming. So again, I welcome you all to this national workshop on Cancer Genomics, Clinical Research, AI Pathways to Oncology Careers. So the first session, as mentioned by Tithi ma'am, is all about the basic foundations and frontiers that is related to cancer. So to begin with, I have an agenda for you. What are the topics or what are the criteria of this session? The very first, I'll be going with the genetic basis of cancer. That means what are the basics of cancer, what are the genes involved in the cancers, what kind of mutations contribute to the cancer. Next, we will look on to the key technologies that are driving cancer genomics. That includes NGS, CRISPR, single-cell sequencing. Followed to that, I'll also give you a brief out on multi-omics approaches in cancer research. Of course, the details about it will be taken up in the upcoming session. I'll just give you an overview. Next, we will also look into some case study about how precision genomics has transformed the treatment of cancer. And lastly, what is the role of liquid biopsies? What are liquid biopsies and how do we use for real-time monitoring? So let's get started with the session. Are you all ready? Yes? Shall I go with the session now? Good morning, good morning. Okay then. So obviously the very first question arises when we get into this topic is what exactly a cancer is all about, right? You all might be already aware about it, but still a brief introduction for that as well. So cancer is basically a non-infectious disease that starts at the molecular level of the cell, ultimately affecting the cellular behavior. And it is not just about a mutation that happens in a single cell or a single disease, but a collection of conditions that are being characterized by uncontrolled growth as well as division of abnormal cells without undergoing any sort of differentiation. And I'm pretty sure that you are already aware about the term, what is a differentiation and what is a division is all about. So division is simply making a copy of itself, while differentiation is all about making a cell with defined functions. So when it comes to cancer, it never undergo differentiation. It simply undergo division, accumulating mutations. So let's understand what are the factors that will contribute to this genetic instability. Now that includes defects in various aspects of biological process. Like we have defects in DNA replication machinery, defects in repair machinery, defects in cell cycle checkpoint mechanisms, mistakes that happens in the mitotic level, and maybe due to the presence of abnormal chromosome numbers, which we collectively call as ploidy or the mutations. So since we know what is a cancer is and what are the factors that contributes to the genetic instability, so definitely there are some unique features which are unique only to the cancer cells. Let's find out what are they. The first includes sustained proliferative signaling. That means irrespective of the presence of growth factors or absence, the signaling will continue. Signaling continues means obviously it leads to continuous cell division. Evading the growth suppressors. That means by default we have our immune system, our cells are equipped with defense mechanisms that can suppress any kind of unwanted things like diseases. But when it comes to cancer cells, they are able to overcome such suppressors. The third important fact is activating invasion and metastasis. That means they are able to enter inside and spread from one location to other. Next, enabling the replicative immortality. That is what? Division, it continues. It keeps on replicating the cell. And then we have inducing angiogenesis. That means they will be creating new blood vessels for their own. They are not depending on any host factors, but they are creating their requirements. And lastly, one of the important features is that they are able to resist apoptosis. In fact, this is one of the major cause why chemotherapy doesn't work. Okay, so that is about the characteristic features of a cancer cell. Now, I'm just giving you a glimpse of how exactly this carcinoma develops. So, to begin with, we have basal lamina present. Basal lamina is nothing but the basement membrane onto which our epithelial cells lies. Below to that, you will find blood vessels present, connective tissue present, as well as lymphatic system. So within this epithelial cells, you can find a single initial proliferative cell is present. Within few days, their division increases. And next, after some days, you will find that a small adenoma is created. Adenoma is nothing but as of now, we call it as the benign because it is not spreading. Then it can reach a particular size where we call it as an intermediate adenoma. And then it becomes a larger adenoma, and after larger adenoma, it becomes carcinoma. So when can I say that this adenoma is converted into carcinoma? When they will start invading our blood vessels and lymphatic system, because it is through these two systems it can easily undergo metastasis, that is moving throughout the body. Okay, so that is how a cancer can develop. Now let's look upon to some of the important genes that are playing a crucial role in cancer. So normally we have proto-oncogenes that is required by our cell for survival as well as proliferation. Like we have the RAS, like we have the anti-apoptotic proteins, those factors or transcription factors that are involved with proliferation, signaling proteins, anti-apoptotic proteins, all this comes under the examples of proto-oncogenes. So basically, they undergo gain of function mutation. That means they have gained some function because of which excessive proteins are synthesized. And these are considered to be genetically dominant. Mainly arises due to point mutation, chromosomal translocation, or maybe amplification. Second set of genes includes the tumor suppressors. As the name indicates, they are responsible for suppressing any kind of unwanted cell survival or proliferation. So the examples includes exactly the opposite category. We will have apoptosis promoting proteins, inhibitors of cell cycle progression, checkpoint pathway proteins, then components of signaling pathways, that means those components that will restrain or prevent the cell proliferation. And these kind of genes actually involves loss of function mutation, because if they are losing their function, then only the cell survival happens. And this is considered to be genetically recessive. Arises mainly due to deletion, point mutation, or maybe methylation. The third set of genes includes the genome maintenance genes. That means those genes that are responsible for maintaining the genes throughout. So whatever the damage happens to the DNA level, we will have a specific DNA repair machineries that will be recruited to the site to carry out the repair. Like you might have heard about base excision repair, nucleotide excision repair, mismatch repair. So they have got their individual set of machineries to carry out the process. And like tumor suppressor genes, they also undergo loss of function mutation, considered to be genetically recessive, and they also arise by same set of deletions, point mutations, or maybe methylation. So the major contributors of cancer, I can say these are the two genes. So let's have some deep understanding on that. See, the thing is that you have to remember one fact. Always remember that proto-oncogenes are undergoing gain of function mutation. That means a single allelic mutation will be enough to convert a proto-oncogene into an oncogene. That's the reason we call it as a dominant mutation. On the other hand, if I take the case of a tumor suppressor gene, suppose the person has inherited normal sets of alleles, but in the walk of life he has underwent one allelic mutation. No problem with that. But if the same person undergoes two allelic mutation, then only this tumor suppressor genes will converted into an oncogenes. And that's the reason we call it as a recessive mutation or a loss of function mutation. So this point has to be remembered very carefully because it should not be mixed up. Gain of function converts proto-oncogenes to oncogenes. Loss of function, these are the mutations that are related to tumor suppressor genes, making it converted to an oncogenes. Next, and one more point, we also see that this proto-oncogenes are considered to be the accelerators of life. You might have come across many times this word. Why we call so? Because as I told you, they promote cell growth and division whenever required. But when mutated, what happens? They will stuck in the on position. So when a cell is present in the on position, it will try to grow uncontrollably. Similarly, this tumor suppressor genes, we call it as brakes of life. So normally it stops the cell growth, repair the damage, or even it will trigger cell death whenever there is a requirement. But when mutated or lost, the brake fails. And you can imagine what happens if the brake fails. So the cell will keep on dividing. I hope so the concept of tumor suppressor genes and proto-oncogenes is clear to all of you. Yes? Completely clear, right? Without any confusion. Okay, that's good to hear. Now let me give you one example. Actually, we have a lot of examples of tumor suppressor genes and oncogenes, but let me give you one example from each of them to understand it better. Okay. So suppose we have a double-stranded DNA that underwent a damage. So if we have normal tumor suppressor genes, BRCA1 and BRCA2, which is considered to be the very well-known tumor suppressor genes, it is responsible for carrying out the repair mechanism. But the problem arises when these genes are mutated. So when these genes are mutated, what happens is that there won't be any repair happening. So it is at that point a checkpoint activation happens in the form of a kinase. So kinase function is to add phosphate to the target. And who is the target here? P53 gene. And P53 is considered to be the guardian angel of the genome. Why do we call as a guardian angel of the genome? Because these are the sensors of DNA damage. Like if any damage happens, the very first gene to recognize it is the P53 gene. So once they are phosphorylated, their level increases. And after that, there are two fates for that damaged portion. At first aspect, it will try to repair it. That is by recruiting P21 genes that causes the cell cycle arrest. Because if cell cycle is not arrested, the mutations will accumulate. So initially they will try to arrest the cell cycle, recruit the repair machineries, and once everything is fine, they will restore the cell cycle. But suppose the damage is too severe, then in that case what will happen? Then there is an alternative pathway where they will produce or activate pro-apoptotic proteins like BAX. Now that will induce the cell to undergo apoptosis. Okay. Now, in the overall picture, everything is fine. But a question can come. What is this MDM2 is all about? Well, MDM2 is a kind of ubiquitin ligase which will keep the p53 in the optimum level, or I can say a low level in the cytoplasm. So MDM2 sequesters the p53 unless and until there is a requirement. So whenever a damage comes, whenever it gets phosphorylated, this MDM2 cannot retain p53. At that time, these two genes will be separated and they are ready to carry out the respective processes. Okay. So this is an example of a tumor suppressor gene. Let's have a look into the oncogenes, or I can say the proto-oncogenes. So you all might have heard about Burkitt lymphoma. Have you heard about this? Thank you, Paris. Yeah, have you heard about this Burkitt lymphoma? No one has heard about it? Okay, I can see some of you have heard about it. Some are saying no. No problem. See, you have heard about a gene known as MYC gene. You know, what is its normal function? They are responsible for regulating the expression of a large number of proteins and non-coding RNAs like rRNAs, microRNAs, tRNAs, which are involved with cell growth, proliferation, as well as survival. In fact, MYC gene is one of the proto-oncogenes that are commonly altered in human cancers, and they are often amplified within the genome or rearranged as a result of chromosomal translocation. Now, just see this case. This is a normal chromosome where you can see c-MYC genes are located where in chromosome number 8. And we have immunoglobulin heavy chain gene which is located in chromosome number 40. So upon chromosomal translocation, what happens? This portion of the gene will be shifted here. And now here you can see rearranged chromosomes. So normally this immunoglobulin heavy chain, wherever it resides, it is responsible for constitutive gene expression. So in that case, if this particular c-MYC gene is also translocated over there, then what happens? They will also undergo constitutive gene expression, right? So in that case, it creates a kind of lymphoma. We call it as a Burkitt lymphoma. Nandini, proliferation means division only. Okay. Proliferation in simple language, it is cell division only. It is just dividing itself. Okay. So this becomes the example of a proto-oncogene contributing to cancer. Next, let's understand what are the key technologies that are driving the cancer genomics part. So as you can see, I have already mentioned in the very beginning under the agenda that we have many technologies that are involved with cancer genomics, but the major ones includes NGS, CRISPR, and single cell sequencing. So let's start up with the NGS first. So under NGS, I have taken an approach, we call it as a comprehensive genomic profiling. Now this approach utilizes a single assay to assess around hundreds of genes, including relevant biomarkers as established in guidelines and clinical therapy for therapy guidance. And this particular approach is responsible for detecting various types of genomic alterations. It could be single nucleotide variants, insertions or deletions, copy number variations, gene fusions, and even some genomic signatures like tumor mutational burden and microsatellite instability. So what are these terms? I will come into details in the upcoming slides. So first let's understand how this CGP, that is comprehensive genomic profiling, is going to help with. Okay. So first what we are having, so whenever we go for any profiling, obviously the process begins with obtaining the respective samples, that is the tumor DNA or RNA, either extracted from tissue biopsies like specific tissues, whether it is a lung tissue, colorectal, breast cancer, anything. or we can go for liquid biopsies. Again, the details of what is a liquid biopsy, I will come into details. As of now, you just remember liquid biopsy, as the name indicates, fluid. So the best fluid in our body is the blood. So blood sample contains some kind of circulating tumor DNA that can be taken. Now this cells will be subjected to CGP. Okay, so what they will identify? They are going to identify different types of mutations, like gene A is mutated in breast cancer, gene B is mutated in lung cancer, gene C is mutated here, gene D is mutated here, and they can also identify some kind of gene fusions also, like NTRK. Now, what is an NTRK? So this NTRK is a kind of genetic alterations where this specific gene, that is a neurotrophic tyrosine receptor kinase gene, they will break down and fuse with another gene, forming a hybrid protein. So this abnormal protein, of course, will undergo uncontrolled cell growth that leads to cancer. And this protein is encoded by a specific receptor known as TRK receptor. So we have some subtypes also present, A, B, C, but we are not going into details. Corresponding to that, we have TRK receptor present. So that's the reason here they have provided with the TRK inhibitor itself. Because if inhibitor is there, obviously there is no point of activation, right? So after that, what happens is that once they are given with the therapy, so each gene mutation will be provided with a specific therapy. Like gene A, they have therapy A, gene B, therapy B. So like that, it goes on. So ultimatum is what? After providing the therapies, you will get two responses. One, we call it as a responder. Now, what is a responder here? Responder is a positive outcome. Positive outcome means the patient will be responding to the corresponding therapy. Second one is the progressive disease. Now that is a negative one because cancer is not able to respond, and that indicates either resistance, and that resistance is something has led to progression. So in such cases, what happens? It should undergo further analysis, and this further analysis can be carried out by immune checkpoints, microsatellite instability, and tumor mutational burden. These are considered to be the markers which will guide for immunotherapy decisions. Okay. Now, once you get the data from the CGP, whether the response or resistance, now they will be fed into the drug discovery pipelines. Now, that will help in identifying the novel targets for cancer therapy, and also it helps in the identification of new drugs for any unaddressed mutations or maybe any kind of resistant tumors. Now, as I mentioned before, let me explain you what is this term TMB and MSI related to cancer. So, NGS analysis for TMB. So, what is TMB? TMB is the total number of mutations per megabase of DNA within a tumor genome. We can say that it measures how many somatic mutations are present within the tumor cell. And this can be done with the help of some sequencing technologies like whole exome sequencing and all. And how we can measure this TMB? Just suppose we take a tumor that is having around mutations, 240 mutations are there, where within a 30 MB exome. Now, how we are going to calculate the TMB? Simply divide it. So, TMB will be equal to 240 by 30. So, what you are getting? You are getting 8 mutations per MB. Right. So, whenever a new protein is produced within a cancer cell, that protein we call it as a neoantigen. So, simply remember, higher the TMB, higher will be the neoantigens. And if neoantigens are higher, obviously they will be presented by the MHC class 1 molecules to be recognized by the respective T cells. Okay. So, what is mentioned here? Let's have a look. A higher TMB suggests that a greater likelihood of producing the neoantigens, which makes the tumor cells more recognizable to the immune system. How? With the help of T cells. CD8 plus, right? And high TMP can be correlated with better response to immune checkpoint inhibitors, specifically in cancers like lung cancer, melanoma, even bladder cancer. So if I have to give you in the form of a flowchart regarding TMP, simply I can say, if there is increase in TMP, it indicates there is increase in the mutations. If increase in the mutation, obviously there will be increase in the production of abnormal proteins. So this can lead to stronger T cell recognition and response. Okay, so that is how the TMP works. Now I have a small graph for you, this TMP for each tumor from TCGA profiles. Now what is TCGA? Cancer Genome Atlas. This was launched in the year 2006 by the US National Cancer Institute and National Human Genome Research Institute. So it's a comprehensive effort. It's a program to understand the molecular basis of cancer. In fact, this organization has profiled hundreds of individual tumors in order to characterize their genetic as well as the molecular basis by analyzing their DNA, their RNA, proteins, even epigenetic changes across number of tumor cells. So when you look into this graph, okay, you can see we have a y-axis present and we have an x-axis present. So y is all about the mutated proteins and x is all about the type of cancers. As you can see here, it is mentioned. Right. Now, every in between you find a black dot. So this is nothing but the median of that particular cancer type. Okay, and median number of mutated protein for the corresponding cancer. Now, looking into this TCGA profile, what you can say? Which one is having the highest TMB? Looking into this genetic profile, which one is having the highest TMB? Highest TMB. Yes, Ayushi, Selvakumar, Tanushri, Sweetie, Priya. Yes, absolutely correct. So why do you choose melanoma as the highest TMB? Because they have the highest number of mutated proteins present. Now, if I say the lowest mutation burden, it will be AML, thyroid, renal chromophobe and all, right? And the middle one will be the moderate one. So where the neoantigen secretion will be higher, obviously I can say that here only the neoantigen production will be higher. The highest mutational loads I can say, thus making it as a top candidate for immune checkpoint therapy because of that, right? So that is about the TCGA profile. Now coming to microsatellite instability. So microsatellites are what? These are 2 to 5 base pairs of DNA sequences that have been randomly repeated from around 10 to 60 times. And these regions are prone to be base pair mismatching at the time of DNA replication. But as I told you already, we have the repair machineries present. So this can be easily corrected by the mismatch repair mechanism or the mismatch proteins. But the problem arises when the repair mechanism is impaired. So at that time, what you will find genetic hypermutability, that is hypermutation, and microsatellite instability results. In fact, we have seen this MSI being approximately 15 to 20% of colorectal cancers. In fact, one study compared the treatment with PD-1 inhibitor, that is a kind of immune checkpoint inhibitor for this colorectal cancer, with high levels of MSI to those without MSI. But with this comparison, what they found is that MSI with high levels showed an objective response rate of 40% and disease control rate of 90%. Now I know a question will arise. What is this ORR and BCR is all about? Well, ORR is nothing. Simply remember it as the percentage of patients. How many patients had their tumor shrink because of the treatment? And BCR is again a percentage of patient whose patients did not get worse. That means they are as it is. It is not deteriorating. It remains as it is. So this is like 40 out of 100. This is like 90 out of 100. So you can imagine the stability. You can imagine the detection ability. Right. So that is about the NGS detection. I hope so I'm clear to all of you this part. Next, we are moving on to, I have some examples also, like a few studies that are being carried out with different cancers. As you can see, for lung cancer, whole genome sequencing was carried out where the focus was the tumor heterogeneity, where they have identified multiple mutations that contributed to drug resistance. So what is the application? They provide insights into personalized treatment strategies. Second was breast cancer. Again here, exome sequencing was carried out where they focused on, of course, the genetic variants related to BRCA1, BRCA2 gene. And what they found? They uncovered the novel mutations that are linked to this cancer, and the implication was that that resulted in enhanced genetic counseling as well as risk assessment. Similarly, you can see a lot of cancers has been studied so far with the help of different sequencing technologies. Focusing on specific part of the cancer, what are their key findings and the respective applications as well. Okay. Now, next moving on to the another important technology, we call it as a CRISPR-edited T cells for cancer therapy. So, you have heard about CAR T cells. Have you heard about CAR T cell? Tumor heterogeneity means, see, tumor is made up of different types of cells, right? It is not about the homogeneity. That means it doesn't mean that it has a single type of cell. So when different types of cells accumulated together, we call it as a heterogeneity. Okay, Priya? Yeah. CAR T cells, which involves taking the T cells from the patient itself and then working towards it, right? Then they are being manipulated in the lab and then they are being re-infused back to the patient. So that is your CAR T cell therapy is all about. Now, how we can use CRISPR with CAR T cell therapy? In fact, the first trial in the US has been carried out in the year 2019 at the University of Pennsylvania, that is CRISPR-made cancer therapy. So it was even partially funded by NCI as well. So it's a type of testing on immunotherapy where the patient's own immune cells are being genetically modified in order to see and kill the respective cancers. Okay, so let's have a look onto that, how exactly it works. So obviously the very first step, since it is CAR T cells, so we have to extract T cells from here. So the very first thing is that the blood will be taken from the patient so that we can get T cells out of it, because T cells, as you know, they play a very important role in immune defense. In fact, there is a term for this extraction procedure. In oncological terms, we call it as leukapheresis. Okay, after that, this T cells were edited in the laboratory. What kind of editing? Editing in the sense reprogramming. So how the editing and reprogramming is done? This is done by 4 major changes. First is that they have introduced NY-ESO receptor into the gene. That is the first change they did. Now, what is this NY-ESO receptor? This is the kind of protein which you will find often being expressed on the surface of many cancer cells, whether it is your myeloma, melanoma, sarcoma, anything. In fact, they serve as a GPS, allowing the edited T cells to specifically recognize and look into the cancer cells that have this marker. The second editing they did was they deleted 3 genes. What are the 3 genes? PDC1, TRAC and TRBC. Now, what is the significance of removing these genes? This PDC1 is usually we call it as a programmable death gene, PD1. You know, when it comes to immune checkpoint inhibitors, okay, if there are no checkpoint inhibitors present, then what happens, you know, this APC and T cells, they interact via B7, PDL1, CTLA4 and PD1 on T cells. But the contradictory fact is that tumor cells can also express the same molecule. So what happens? The T cells will be inactivated, which induces the tumor growth. On the other hand, how you can overcome this? You can overcome this with the addition of checkpoint inhibitors like PD1 inhibitor and PD-L1 inhibitor. So if you are adding this inhibitors, what happens is that they won't be interacting with each other. If they are not interacting with each other, T cells will be activated. That will help in the treatment. Okay. So in this case, what they are doing is that they are removing that PDC1 cells. So your the T cells will not be activated. Secondly, what they are doing is that they are removing TRAC and TRBC. Now these are T cell receptor alpha and beta chain genes. So what they are literally doing is that they are trying to remove, like they are manipulating in such a way that they will code for the T cells natural receptor. Okay. So once you are deleting these genes, what happens is that it will prevent whatever the new NY-ESO we are inserting, it will prevent this receptor from mixing with the old ones. So it makes it more effective. And at the same time, it also reduces the risk of graft versus host defense, where the infused cells has the chance to attack the patient's own healthy cells, right? So in order to avoid these things, they are eliminating these 3 genes. Now next what they are doing? Next they are multiplying them or growing them in this edited version of T cells in the laboratory. So you will get CRISPR edited T cells which can be infused back to the patient. So this is basically an autologous transplant because you are taking the cells from the patient itself, manipulating and re-injecting them back. But at this junction, you know, the patient undergoes a brief course of chemotherapy. We call in the oncological terms, we call it as a lympho depletion. Okay, now what does this mean? Before the infusion, this ensures that even if any immune cells are there, they will be removed and they are creating space for the newly engineered cells in order to expand and supply. Okay, so after this, what happens here? CRISPR edited T cells has already been infused here. Now they have circulated throughout the body. They have the receptors. So wherever they will find this corresponding markers, they will be able to identify it. They will activate the T cells and they will kill the cancer cells. Okay, so this is how the CRISPR edited T cells is going to work. Next, we have the single cell sequencing. So what is a single cell sequencing all about? As the name indicates, it studies the genetic material of individual cells rather than analyzing the mixture of different cells together. Like you might be aware that in traditional sequencing methods, signals from thousands or millions of cells are being averaged. So this will mask the difference between the cells. But when it comes to single cell sequencing, it basically examines the cell individually, revealing cell to cell variability when it comes to gene expression or mutation or maybe epigenetic patterns. Now, let's understand how this single cell sequencing is being utilized in cancer. So we have tumor microenvironment present. So tumor microenvironment doesn't mean that it only contains tumor cells. It will contain plethora of cells, endothelial cells, fibroblasts, pericytes, adipocytes, immune cells. All these cells will interact with the blood vessels and influence tumor growth, immune evasion, as well as drug response. So with the help of single cell sequencing, they will allow profiling of all these cell types in a single level. Second is the cellular heterogeneity. As I told you, different populations. So tumors are not uniform. They consist of different subpopulation of cells. And each cluster will have their unique set of genetic as well as transcriptional features. For example, I say cluster 1 is all about those genes that are more aggressive and therapy resistant. But if I say of cluster 2, they are having slower growing or therapy sensitive cells. So with the help of the single cell sequencing, what happens is that they will identify these differences. That is the major setback, major thing over here. Next is the individual cells gene expression profiles. Like we can go for transcriptome profiling. Transcriptome means what? Where we are trying to capture the gene expression profiles of individual cells that are present within the tumor. And here you can see a map present where there is up-regulated and down-regulated genes are being identified. So that is happening mainly because of this single cell sequencing technology. Apart from that, they can also undergo further analysis by pathway analysis and all, where they can cluster them into two types of cells, where they will be able to identify which genes are being shared, which genes are unique, which pathways are driving the tumor progression or causing the immune evasion. So you will also find there are some genes which are overlapping to each other as well. And the last part is one of the impactful application over here is identifying treatment resistant and treatment responsive clones. So this single cell RNA sequencing, they will detect transcriptional signatures that are associated with resistance and also predict why certain cancer cells can survive the therapies and why they are dying. So this will help in designing the precision therapies to overcome the resistance mechanisms. Okay. So these are the three major technologies when we look for cancer genomics. Now let's look into the workflow of integrating the omics data when it comes to cancer research or clinical applications. So obviously, in this case, you have to use a number of bioinformatic tools. Then only you will be able to analyze, integrate and interpret the necessary data so that it will give you with better understanding of cancer biology. Not only that, it will also help you to develop the personalized treatment strategy also. So obviously the process begins with collecting the biological samples from the patient. So it could be a tissue sample or a sample. So if we go for genomics, we can go for a whole genome sequencing or a whole exome sequencing. So if we are capturing all the genetic variants, that goes for genomics. If we are specifically focusing on protein coding regions, of course the exome sequencing. Then for interpretation, for analysis, they can detect if there is any single nucleotide changes are present in the DNA level. They can identify if there is any frame shift present, that means small insertions or deletion. They can identify if there is any copies present, number of copies of a specific gene is present. They can also determine if any kind of structural changes has occurred, whether it is translocation, inversions, deletions, duplications, anything. Then coming to the transcriptomic level, that is all about the RNA level, right? So it can go for differential expression. That means comparing the gene expression levels between two different conditions or samples. Like for example, I can take a normal cell to that of a tumor cell, and that indicates a very good example of differential expression. Next, identification of gene fusion. That means there are some genes which are often found to be fused with each other in certain cancers. If you remember, as I told you with respect to Burkitt lymphoma. Then alternative splicing, analyzing different ways that RNA can be spliced in order to produce different variants right from a single gene itself. So different exon combination that will be leading to diverse protein isoforms. And the last part is the RNA editing part. That means we are modifying the RNA sequences that is responsible for altering the encoded information, but it is not changing anything in the DNA level. in this technology will be the epigenomics part. Now that includes bisulfite sequencing and chip sequencing. Bisulfite sequencing specifically focusing on studying about the DNA methylation pattern. And you all might be aware that methylation is considered to be one of the important epigenetic trait with respect to the gene expression part. Chip sequencing is also known as chromatin immunoprecipitation sequencing. They are mainly involved with mapping protein to DNA interaction. So what is the protein we will find? Obviously, we will find histones over here, right? So analyzing the chemical changes to the histones that can contribute to the remodeling complexes and all. Then, of course, identifying where the transcription factor binds to the DNA obviously will be influencing the gene expression pattern. Now, once you have analyzed all these things, what will be the next step? You have to collect the information from all this analytical part and interpret it, or bring them together. So for that, by that, in fact, I can say that they will provide you with the information about functional effect of mutation. That means understanding how a specific mutation is affecting cellular functions and contributes towards cancer. Secondly, you will be also able to identify or map any kind of genetic or protein interactions, the pathways that are commonly related to cancer. You might be aware that most of the times RAS pathways are mostly involved with cancer, the PI3K pathway, the MAP cascade and all. Then integrative analysis, that is combining the data from multiple resources, you will get a comprehensive overview of cancer biology. So what will be the ultimatum here? The ultimatum here is to translate these findings into clinical applications. That would be in the form of targeted therapies, improved diagnostics, or even personalized treatment strategies as well. Okay. So that is about the omics approaches. Now coming to the precision oncology part. Now precision oncology means what? Precision medicine means what? Based upon your genetic profile, how we are creating the treatment for that particular individual. So when I say precision oncology, here are some key components to be focused on. The very first thing is the diagnosis part. That is it a cancer? If so, what kind of cancer it is? So this indicates what? Accurate diagnosis. So this accurate diagnosis takes the first step in precision oncology. Identifying what kind of cancer it is, identifying the molecular feature of that cancer. Now this will ensure selecting the most effective treatment. Second is the risk assessment. Does this patient have a higher risk? That means the risk factors in the sense genetic predisposition, the lifestyle change, the tumor stage. So all this will determine the aggressiveness of cancer and the likelihood of recurrence. Third is the prognosis part. That means how serious is this cancer and what are their survival rates. So understanding this prognostic biomarkers, that means the tumor grade or the genetic mutation, will provide us with better insights about their progression. Next we have pharmacogenomics part. Pharmacogenomics means what? How a patient's genetic makeup can affect their response to drugs. So this will help in optimizing the drug dosage, thereby minimizing the side effects. Thirdly, what is the best treatment and will this cancer respond to the treatment or not? See, precision oncology is all about using molecular profiles. That includes the genomics part, the proteomics part, in order to identify targeted therapies or immunotherapies that are likely to be effective in the patient's specific cancer. And the last part is the monitoring part. That is, how is this cancer responding to the treatment, or is it relapsing or not? So regular monitoring through imaging techniques or biomarker techniques becomes an effective tool to find out the effectiveness of treatment, at the same time to check if there is any signs of relapse. Okay. So that is a basic introduction to the precision oncology. Now this is a comparison image of traditional medicine to that of an innovative personalized medicine. So what is the major difference, in traditional medicine, we always see that one size fits all approach. That means if 5 people are suffering from colon cancer, they will be provided with the same treatment regimens, the same general guidelines, which is not being tailored for their unique biology. So even though they are having different biomarkers, they will be given the same treatment only. So what will be the output? The output will be different. Like it could be effective for some people, others may not benefit at all, and some people may even experience harmful effects as well. Now, when I compare it with the personalized medicine, for example, the same colon cancer, if we take, we go for personalized therapy based upon the type of biomarkers that are being expressed on their cell. See, biomarkers can be anything. It could be molecule, it could be protein, it could be gene, anything. So, based on that, it is being designed, right? So, it's like a tailored therapy for the treatment is being customized based upon the requirement. So, in that case, most of the times you are going to get the positive effect only. That means higher likelihood of the treatment success due to the targeted therapy, or we can say that they are having very less adverse effects compared to the traditional ones. Now, here are some few real life examples of real life case studies that showcase the remarkable success of precision medicine in the field of oncology. The very best example is the Glivec in CML. It is the prime example of precision medicines. In fact, the first targeted therapy to be approved by FDA. Now, what happens here? Here this Glivec is targeting a gene fusion. We call it as an BCR-ABL fusion that is responsible for causing CML. How? Have a look. So, suppose in a patient, this BCR-ABL gene is active. So, once this gene is active, ATP hydrolysis takes place. That phosphate will go and bind to the substrate protein, and this will help in the cell proliferation and survival, resulting in leukemia. Now, what is the role of Glivec here? The Glivec will go and bind to the ATP binding site. So, what happens? ATP hydrolysis won't be taken place. There won't be any hydrolysis. So, substrate protein won't be activated. So, what happens? No signal, no leukemia. So, this is one of the best example of this studies, the case studies. Apart from that, this BCR-ABL gene, usually we call it as a Philadelphia chromosome. You can see the ABL gene shifted to BCR, the shortest chromosome, we call it as a Philadelphia chromosome. Then we have Herceptin in HER2 positive breast cancer. So this tends to be more aggressive, and Herceptin is a kind of monoclonal antibody specifically targeting this receptor. So when we are using this receptor along with the chemotherapy, it has been incredibly effective in increasing the survival rates, thereby reducing the risk of recurrence in these patients. Then we have the larotrectinib, which is involved with NTRK fusion positive tumors. So this is a targeted therapy developed to treat tumors with this NTRK gene fusion. They also provided with impressive results, with some patients experiencing complete responses even in advanced stage cancers. Keytruda in immunotherapy. So this is a kind of immune checkpoint inhibitor, and it is known to be a game changer in oncology. You know what it will do? It blocks this PD-1 or PDL-1, the one which I had told you through an image, that the immune checkpoint inhibitors, they will directly target these inhibitors. So what happens? The T cells will be activated. Usually with the presence of these PD-1 and PDL-1, what happens is that T cells becomes inactivated and tumor growth takes place. But if this is inactivated, what happens? It's a huge success. So in certain cases of melanoma and lung cancer, Keytruda has shown advanced and previously untreatable cases with long-term survivors. Okay, so these are some real life examples. Now moving on to the last part of this session, that is liquid biopsy. One of the trending topic, one of the trending technique when it comes to cancer. So liquid biopsy, as the name indicates, it refers to the process of analyzing a sample of biological fluid. Now the goal here is to identify early signs of cancer, monitoring the progression of the disease, thereby assessing the effectiveness of the treatment. It has become increasingly popular in cancer research and treatment and is expected to impact personalized medicine significantly in the upcoming times. So this is a small comparison of pre-liquid biopsy era to that of a liquid biopsy era. In pre-liquid biopsy era, what happens? No doubt screening takes place. We have imaging techniques like CT scan, MRI and all will happen. Diagnosis will be taken, tissue biopsy, but usually what are they? These are invasive techniques. Okay, and limited repeatability. Then based on that, treatment is being selected, but there is no de-escalation strategies, no real-time monitoring. Simply they will monitor and follow up. That means they will directly, based upon the imaging techniques, they delay the detection of recurrence and resistance. So it will be a late process. But when I speak of liquid biopsy, screening is there, diagnosis is there, treatment selection is there, and the major part is that we have de-escalation strategies and MRT monitoring. So what is this MRT monitoring? Minimal residual disease monitoring. Now that refers to the tiny number of cells that retains in the body even after the treatment. Suppose a person has underwent chemotherapy, surgery or anything, but there are high chances that certain cells will be retained. Now that can be detected by this liquid biopsy era. And de-escalation is nothing but reducing the intensity, duration or maybe a dosage of therapy once this MRD shows the cancer burden is failing. Okay. And coming to real-time monitoring, yeah, that also can happen when it comes to a liquid biopsy. And lastly, of course, they will also go for monitoring and follow-up. Now, next what we are having, this is an overview of liquid biopsies with their application. So we have to take any of this biological fluid. We can take saliva, we can take urine, blood, even breast milk. And what are the components we will get in liquid biopsy? We can get circulating tumor cells. That means these are the intact cancer cells that are freshly coming out of the primary tumor or maybe metastasis and they have entered into the bloodstream. Circulating tumor DNA. These are the small DNA fragments that are being released into the blood specifically by the tumor cells. During apoptosis or maybe necrosis or something like that. Then we have the cell-free DNA, that is DNA will be present in fragments in freely circulation in the fluid, in the blood. And it can be released from anywhere. It could be from normal cells and the tumor cells. Then we have exosomes, that means a kind of tiny sac structures. These are considered to be extracellular vesicles. And how are they being applied? They can be applied in different aspects. For example, they can detect the methylation and histone modification patterns, that is what we call as epigenetic changes. Then they help in identifying specific mutations when in oncogenes or tumor suppressor genes. Always provides for personalized treatment. They also monitor resistance mechanisms in order to adapt to that particular treatment. Then they can classify cancers based upon the molecular features. And the last part is the mapping part, where it involves comprehensive analysis of tumor-specific alterations that happens in the gene level. Now, let's look into the different types of liquid biopsy markers here. The first one is the CTC, circulating tumor cancers. So obviously this image in the first part, you can see how the cancer cells are spreading from a primary tumor site and entering into the secondary site. So first it will be invasion, right? They will try to invade the surrounding tissues that can be facilitated by some cell adhesion molecules like integrin. Now from here they will enter into the bloodstream, that is what we call as intravasation. And from the intravasation, from this circulating tumor cells, they will be traveling, that is extravasation, that means they will move on to the distant sites, and they may exit the bloodstream also, and then they can establish a new secondary site, that is what we call as colonization, often forming micrometastasis or pre-metastatic niche. Now, what is this micrometastasis? These are early stage tumors. And pre-metastatic means that is being already created, a microenvironment that is already present over there. Okay. Then how we can separate this? We can separate it, the circulating tumor cells in different ways, like based upon the cell size, we can differentiate it. Based upon the density difference, we can segregate it. Then it can also use microfluidic devices in order for isolation, or you can also go for filtration to capture large cancers. After that, it can be subjected to techniques like flow cytometry that will identify and quantify the cells based upon the markers. Then EpCAM enrichment method, that means detecting the CTCs with EpCAM. EpCAM means epithelial cell adhesion molecule. Then one can go for immunofluorescence, and then the cell search system, that is a clinically validated system for detecting and carrying or counting the number of CTCs. So once we have detected it, the application goes, we can go for diagnosis, we can go for disease prognosis, we can monitor the treatment, and we can also go for this prognosis part as well. So that is about the CTC as the biomarker. Next, we have ctDNA as the biomarker. So usually what happens, the ctDNA, they will enter the bloodstream through these three processes: necrosis, apoptosis, and exosome. So necrosis and apoptosis, the basic difference is that this is a kind of programmed cell death which is involved with a number of caspases, but this is a general cell death that will spilling the contents outside and all. Exosomes, they are usually released via some extracellular vesicles. So initially they will form early endosome, then they will mature, giving rise to multivesicular endosome, and then they will fuse with the lysosomes to carry out the degradation process. After that, they will undergo, once they are in the circulation, they will carry out the molecular signatures of cancer. That is the mutation and modification part. So it can carry out mutations in the form of DNA mutations, point mutations, insertions, deletions, and methylations like promoter hypermethylation, genomide hypomethylation, any kind of abnormal methylation, mutations in reader proteins, decrease in the acetylation part, then Sweissnap loss of function. Now that is going to create a huge impact on chromatin remodeling complex, then abnormal chromatin structure. And finally, we can detect it through different techniques like RT-qPCR, NGS, droplet digital PCR, even microarrays. Then we have the TEPs. Now, what is this TEPs all about? TEPs are nothing but tumor educated platelets. Okay, so these are blood platelets that undergo functional and molecular reprogramming specifically in response to signals from the tumors. So these interactions will enable the platelet cells to help in cancer progression, metastasis, even the immune evasion. And at the same time, they also serve as a potential biomarker for cancer detection and monitoring. So the key elements In this case, we have the circulating tumor cells, that extracellular vesicles, platelets, the DEPs, non-coding RNA, circulating tumor DNAs. So basically, these components will be released into the bloodstream via different procedures. And after that, each of these fragments can be analyzed by different techniques. If we are taking the DNA fragments, we go for DNA sequencing. RNA fragments mean RNA sequencing. Proteins means we can go for this liquid chromatography or mass spectrometry method. And the technological advancements when it comes to liquid biopsy analysis, we have these many techniques that can be utilized. NGS, dPCR, and every method has their own set of advantages and limitations and their sensitivity and specificity factors. So depending upon the source, depending upon the type of cancers, we have to decide. So that's all from my side. Actually, I have kept a short video animation over here just to show you how that comprehensive profiling happens. So I just want to take permission from Priti ma'am. Can I show this animation, ma'am? Is it fine?
Sure.
Okay, just give me 1 minute, students.
Divya ma'am, it is not audible.
It's not audible.
No, I think you have to share the sounds also. In Zoom setting only, ma'am, you will get.
Can you please tell me, ma'am, where exactly? Actually, I'm not getting it.
The control that you will get on top of your screen, right? So there, those 3 dots will be there. Maybe check that.
This is transcript part actually.
Anything with sharing option? You have to share the sound also. Share the audio. Yes, Suraj, everybody who are joining, you all will be receiving the recording as well. But it will take us some time to, process it and send it across to you. So within a week, you will receive the recording.
Just check if it is audible. Is it audible?
No.
No, I'm not sure, ma'am. Actually, I tried, but it's not working.
For sharing option, okay, so can we do one thing? Yeah, can you send me the link of it, Divya ma'am? Then let me try from my end.
Please check, ma'am. I have sent you.